Welcome to Biopsych, PSY 351. In this module, I'm going to cover nerve cells and impulses. I'm going to cover cells of the nervous system, neurons and glia. I'll talk about the blood-brain barrier, which is an important function of some of the cells of the nervous system. And I'm going to talk about how neurons get fed. I'm also going to talk about the nerve impulse. This is probably the most difficult part of the entire course, so make sure you're prepared to invest some time in understanding this. If you do, you'll be in fine shape, but it's going to take a little extra time, more so than maybe learning about schizophrenia later. I'm going to cover the resting membrane potential, the action potential. I'll talk about propagation of the action potential, the myelin sheath and what it does, and I'll cover local neurons in a bit. This may not make much sense right now, but hopefully after the lectures it will. In this lecture, I'm going to cover cells of the nervous system, and that'll include both neurons and glia. I'm going to go over kind of a rough overview of their structure. I'm going to talk about the function, the different types of neurons, and same with glia. After you watch this video on cells of the nervous system, I want you to be able to describe the basic structure and the function of both neurons and glia. Now, as the book points out, you've got an awful lot of neurons. The cerebral cortex has 12 to 15 billion neurons. Cerebellum alone has 70 billion neurons. Even your spinal cord, which most people think of as just a pathway, a set of wires to carry information from the brain to the periphery and from the periphery back to the brain, has about 1 billion neurons. So we're talking about an awful lot of cells. Now, when we talk about neurons, this is only about 1/10 of the number of cells in your body. So in terms of, not your body, I apologize, your brain. Your brain has 10 times as many glia, the support cells, as it does neurons. So here we have 120 to 150 billion glial cells in your cerebral cortex. Again, that's an awful lot of cells. If you're not familiar with the basic components of an animal cell, you might want to go back and review. I'm going to go over the major ones quickly, and I'll highlight the ones you absolutely need to know. You do need to know the mitochondria. The mitochondria are small little organelles. An organelle is a small substructure within a cell. These provide energy. Okay, so you need to know that the mitochondria provide energy. Things that require a lot of energy, like neurons, are going to have a lot of mitochondria. Other cells, like fat cells, adipose tissue, has very little energy requirement and consequently has fewer mitochondria in the cell. I probably should have actually just started that this cartoon represents a typical animal cell. We're going to have a bunch of components inside. All this stuff inside is going to be called the intracellular. This will be the extracellular, the stuff outside. It's going to be surrounded by the cell membrane. You can think of this almost as a balloon that separates the external environment from the internal environment. All the organelles I'm going to talk about, like the mitochondria, are intracellular. There'll be some later on when we talk about Alzheimer's where there's going to be some important extracellular components. You need to know the nucleus. The nucleus contains DNA. And again, hopefully you remember what DNA is. DNA contains the instructions. One thing I want you to be sure to know is DNA carries the information for the cell to make proteins. I'm going to talk about this a bit more detail later. Okay, but your genes, which are contained in the DNA, contain the instructions to make proteins. So if something's not a protein, you don't have a gene for it. So people talk about addiction. Is there a gene for addiction? Well, is addiction a protein? No, it's not. So there's no gene for addiction. Now, are there genes for cell components that affect how cells function, and that those, when they function abnormally could lead to addiction? Absolutely. Okay, so it's important. I want you to remember this phrase. DNA codes for proteins, okay, and proteins do things. If there's no protein, if it's not a protein, there's no gene for it. Okay, the DNA is contained within the cell's nucleus. All the cells we're going to talk about are going to have nuclei. We'll talk a little bit about ribosomes. Ribosomes make the protein. Okay, so the instructions are going to be contained in the DNA. That DNA is going to eventually send that information out to the ribosomes, which are basically the factories that make proteins. Like I said, proteins do things. I'm going to talk a lot later about enzymes, receptors. Those are all proteins. Okay. So those are the main things you need to know. Plasma membrane, nucleus, mitochondria, and ribosomes. Here is a transmission electron photomicrograph, and I'm going to show you some of these. The reason I bring this up again is there's a couple structures I really want you to know. One is going to be, of course, the nucleus, right? We already talked about that. The plasma membrane, that's also called the cell membrane. So sometimes I'll say plasma membrane, sometimes I'll say cell membrane, same thing. Right? That's going to be basically the latex of the balloon, right? That separates inside from outside. The plasma membrane is going to be really important when we talk later about electrical potentials. Mitochondrion, it's one mitochondrion, a bunch of mitochondria. Okay, these are going to have produce the cell's energy. Endoplasmic reticulum I didn't really talk about too much, but it's a series of membranes that are associated with ribosomes. Now you know what ribosomes do. Remember, these are going to make protein. Okay, so the endoplasmic reticulum, if it has ribosomes on it, is going to be important for making proteins. Now, a neuron has a specific definition. Okay, so obviously a neuron is a cell. Not all cells are neurons. Okay, but a neuron is a cell that's specialized to receive information and then transmit that information to other cells. Okay, so many cells in the body receive information. There's some cells that will transmit information, but only neurons really do both. Okay. Now there's a couple famous names you need to know. One is Ramon Cajal, the other is Camilla Golgi. Both these guys were active in the late 1800s, early 1900s, and they shared the Nobel Prize in 1906. To make a long story short, Golgi basically developed a stain. He developed a technique that allowed us to study individual neurons. Cajal used Golgi's technique to show that your nervous system was comprised of independent cells. Up until Cajal's time, we didn't know whether the brain was basically just one giant cell or that it was made up of many discrete cells. Okay, so Cajal used Golgi's technique to show that. When I talk about methods, I'm going to spend some time talking about Golgi stain. You'll need to be able to recognize tissue using that's been stained using that technique. Now neurons have many different shapes and many different functions. Okay, all of these neurons here are stained using Golgi's technique. Okay, so it's called Golgi stain. To be a little more fancy, it's a silver impregnation method. Basically, what it does is it fills with black about 1% of cells. Okay, and more specifically, not just any cells, 1% of neurons. Okay, so here we're going to learn about the structure, but here you can see the cell body. You can see a bunch of processes coming off of it. One of these is going to be an axon, the rest are going to be dendrites. Okay, you can see this particular cell body has a whole bunch coming off. And you can see the whole cell the cell body as well as all the different branches are stained. Okay? Now here there's one. I told you it's about 1%, right? So in this field of view, even though only one neuron is stained, there's actually 100 neurons there. Now if there's 100 neurons, how many glia are there? 10 times as much, so about 1,000. So this very nice clean section, little piece of brain tissue, shows one neuron. There's really thousands of cells there. Okay, it's just a technique fills this one neuron. Now the nice thing is it completely fills it, so I can study this neuron in great detail. Now this one has many different processes coming off of it. It's very different than this one. Here's the soma, the cell body. Here there's only two processes coming off of it. Okay, and actually this is going to lead to the most basic classification of neurons. We're going to have neurons where we have one, two, or many processes. Okay, so basically you look at the cell body and you count the number of things coming off of it. If it's got a bunch of them, we call it a multipolar neuron. Here in this case, how many are there? Two. So what do you think we call that? Yeah, bipolar. Here in this cell, here's the cell body, right? And here you can see one process coming off. Now it splits, but there's only one process coming off. That makes this a unipolar neuron. Almost all the neurons in your brain are going to be multipolar. Bipolar, we're going to talk mostly in sensory systems. So we'll talk about these in the retina and then in the cochlea. Whoops, sorry. The cochlea. Unipolar neurons are going to be sensory neurons. They're going to carry sensory information to the central nervous system. Okay, so if something's a unipolar neuron, you know it's going to carry sensory information from the skin to the brain. Okay, if it's multipolar, you can't tell me much about it. Okay, but for this course, if it's unipolar, it's a sensory neuron. Bipolar, you're going to be thinking about the retina or the cochlea. Everything else will be multipolar. Now here, we can also use this Golgi stain to look at changes as things develop. So here is some brain tissue. This is going to be the prefrontal cortex. We're going to talk a lot about prefrontal cortex. And we can look at this Golgi stain between a newborn and a 5-year-old. And when you look at this qualitatively, what's the difference? Well, if we look here, we see cell bodies. Okay, some are in better focus than others. If we were to go through and count the number of cell bodies between the newborn and the 5-year-old, we won't really see that big a difference. By the time a human's born, most of the neurons are already there. Okay, but what is the difference? If we look at this neuron, say, I haven't drawn on it yet, so it's easier to see. If we look at this neuron, how complicated is the branching pattern? Well, not too much, right? Here, you can see that there's a lot more branches. Okay, it's going to turn out that every neuron has a whole bunch of these branches. These are called dendrites. Dendrites collect information. They'll have one axon. Axon is going to send information. Okay, so the difference between this neuron and this neuron, this in the 5-year-old is going to be able to collect a lot more information. Okay, so one of the things when we talk about development, I want you to remember, by the time you're born, you've got about all the neurons you're ever going to have. What changes is not the number so much, is the relative branching of them. Right, and I already went through this a little bit for you, but this is a classification of neurons. Here, again, we're going to look at the cell body, and we're going to count the number of things coming off the cell body. How many do we have here? One. So this is going to be a unipolar neuron. That I want you to remember is going to be sensory. Okay? Over here in these neurons, although they're different shaped, what are you going to see? Well, if we look at the cell body and we count the number of processes, okay, a process is just something coming off of it. You can see we've got a whole bunch. Okay, so that's going to make this multipolar neuron. If there were only 2 processes coming off of it, again, it would be a bipolar cell. Now, as far as a neuron goes, I want you to know the dendrite, the soma, the nucleus, the axon hillock, the axon, and myelin. Okay. Sorry, I had a hiccup there. The node of Ranvier, and then the terminal bouton. And I'll show you where all of these are. One thing, I apologize, this shouldn't be capitalized. Only proper nouns should be capitalized. Okay, so hillock is not a proper noun. Ranvier, like Golgi, is named after a person, so the R should be capitalized when you write it. Now, in a multiple choice exam, obviously this isn't going to matter too much, but whenever you correspond, you write, you talk about, it's important to know what's capitalized and what's not. It's one of the ways people know whether or not you've been well trained. Alright, so this is a checklist of the structures I want you to remember. Here's a cartoon, and here's where all those things are. So the soma is going to be the same thing as the cell body. Okay. The soma is going to contain the nucleus. I already told you that the nucleus contains the DNA, right, which are the instructions for proteins, right? The ribosomes are also going to be in the soma, right? Now, there's a whole bunch of different processes. These are going to be dendrites. Dendrites are going to collect information. If I have more dendrites, I collect more information. Okay. The axon hillock is a special area that's right here. This is where the nerve impulse, sorry, this pen, I'm not used to it yet. This is where the nerve impulse begins. Okay, so I'm going to talk about the action potential later. Remember, the cell body and dendrites right now don't have action potentials. I'll talk about exceptions later, but for now, in a typical neuron, no action potentials here. The action potential starts in the axon hillock, and it gets carried down this axon to the end of the neuron. Okay, at the end of the neuron, we're going to have the terminal boutons. I call them boutons. If you want to be cool, you'll call them boutons too. I'm joking. A lot of people call them buttons, but in the neuroscience field, I've only heard them called boutons. Okay, the presynaptic terminals are the same thing as the terminal buttons and the same thing as the terminal boutons. Okay, so 3 different phrases for the exact same structure. Coding the axon, okay, which is going to be this one bit of cable here, we're going to have myelin. Okay, myelin is basically going to be insulation, and it's made up of fat. Now, in between beads of myelin, this is where we have that node of Ranvier. All right, and so I'll talk more about these structures later. For right now, just memorize what they are and where they are, and in real general terms, what they do. One thing about all neurons have one axon. What's different is how many dendrites they have. Okay, so one axon. You can have none or many dendrites. Okay, so multipolar neuron, if I tell you it's got 50 processes coming off the soma, how many axons does it have? 1. And only 1. Now here's a picture of what kind of a neuron? Again, soma, right? Here. got one process coming off. This is going to be unipolar. Right? And this is a good figure to help you remember. Here we've got the skin. Something's going to touch the skin. Could be heat, could be pressure, could be vibration. That's going to stimulate these sensory endings in the skin. These are then going to send a signal from your skin down this axon into the spinal cord. From the spinal cord, it's then going to get carried up to the brain. We'll talk about these sensory pathways later. Okay, but all of these sensory neurons are going to be near the spinal cord. That's where the cell bodies are, and they're going to carry information from the skin into the central nervous system. Now, I talked about dendrites. Okay, so dendrites are going to be fairly large dendrites, or large structures. So all of these kind of yellowish things are going to be dendrites. Off these dendrites, you're going to be able to see these little orange things. Okay, so these little orange things are the spines. We're going to talk more about synapses. Okay, but synapses are the connection between neurons, where one neuron transfers information to another neuron. Most of those connections are going to occur at dendritic spines. Okay, so the more spines you have, the more synapses you have. Okay, so remember those dendrites, right, were fairly large. If we look back here, right, these are dendrites. Off these, you're going to have little tiny, bumps, and those little bumps are going to be the spines. Now, a quick little bit of terminology. We're going to talk about afferent and efferent neurons. Okay, afferent is going to carry information to. Efferent is from. Okay, again, sorry, that shouldn't be capitalized. Okay, afferent is to, efferent is from. So if I send sensory information from skin to the central nervous system, okay, and then here I'm talking about spinal cord, right? And from here, we're going to go to the brain. Okay, so skin to the central nervous system is going to be carried by afferent fibers. Okay, so the skin receptors, afferent information coming into the spinal cord. Now, likewise, if we want to move, right, in the spinal cord, we're going to send motor information from the spinal cord to the muscles. Okay, so if I'm talking about the spinal cord, I'm going to carry efferent fibers, efferent information from the spinal cord to the muscles to indicate movement. Okay, and the acronym I learned is SAME, sensory, afferent, motor, efferent. Okay, so SAME. Okay, so sensory information going to the spinal cord, afferent fibers. Information going away from the spinal cord to the muscles, efferent information.